Good morning, everyone. I am Dr.R.Carlton from the Department of Environmental Sciences, Bishop Weber College. Today, I would like to uh, talk about on the topic on biodiversity conservation, in specific about the exit to biodiversity conservation strategies. So, first of all today, what is conservation? To have a overview, we will start with what is conservation. So mainly the to conservation is a method where we preserve the genetic or ecological or habitat everything and to restore or to our life support. And what type of life support? So why we need to conserve? We need nowadays require many resources like water, air and soil for mainly for our survival. Water and air, which are one of the basic important thing for a life to survive, and to evade environmental disaster. So why we need to conserve is because of this, because nowadays we face the global warming, climate change and lot of pollution happening. And other thing is to achieve social, economical and healthy well-being of the human life, and which will lead to a sustainable practice and living. So how we conserve? There are two types of conservation. One is in situ and ex situ. Today we are going to see about the second one, which is called the ex situ conservation. So which is an off-site conservation. Conservation away from its natural area. So why we need to go for off-site conservation? Some of you might have questions. So because to avoid certain disaster, conserving at off-site conservation, that is ex situ conservation, can be in the form of zoos, botanical gardens, aquarium, seed banks and gene banks for many living things, and which this ex situ conservation also protects the endangered species, and in an enclosed environment where if a natural calamity, sudden calamity happens, we can try to reintroduce or reincorporate into our environment. And ex situ conservation and purpose, the main purpose is, first one is the rescue of threatened species and produce material for conservation of biological research. So where bulk of germ plasm, storing of germ plasm at various forms of ex situ sites, and supply supply materials for various purpose for even to the environment. So where we can grow certain plants at an ex situ conservation site like a botanical garden or nursery, where it can be again replanted into the environment, growing this and make available material for conservation of education and display, and which will also, this ex situ conservation will try to incorporate the reinforcement and habitat restoration and management. So ex situ conservation, protecting species outside of its natural habitat, as we said. For plants, if you take, like there are different methods of conservation. Few methods, the methods are seed bank, which is cryopreservation, where we store the seeds in a and very sub-zero temperature, and community seed bank, storing it in community in an environment, and gene banks, so which is available for genetical, and botanical gardens and arboretum. So botanical gardens, as we see, we go and see a lot of plants, flowers and varieties present in botanical garden. Arboretum is one thing where we cultivate or we inhibit specific type of plants which doesn't grow in that local environment, and we preserve it in an enclosed area. And biotechnological aspect, we have in vitro ex situ conservation, and for animals we have the gene bank, which is also called as cryopreservation, and storing the genes or DNA in an and very low temperature. And there are other methods where we can do ex situ conservation, interspecific pregnancy methods, where we try out interspecific options, and aquaria and zoological gardens and in vitro preservation. And in ex situ, one of the main focuses is through legislation. So for in situ also this legislation plays a vital role, but it comes under the ex situ conservation because we enforce certain strict laws to the local people or community or to the forest. And also ex situ conservation involves awareness. So awareness is one method where it need to be created at all levels, like from the grassroots level to the shoot level or the top level. So for plants, the conventional methods, the seed banks or the cryopreservation seed banks and botanical gardens and everything are there. So seed banks, what are seed banks? They are cryogenic laboratory facilities for certain species which can be preserved for more than a century without losing their fertility. It can be also used to refer special type of arboretum where seed harvested and crop is rotated. Cryopreservation, so is where we store seeds, organelles, cells, tissues, and organs in a temperature and moisture controlled environment. And if you see the temperature, we store it in the minus 80 degrees Celsius as using a solid carbon dioxide and minus 196 degrees Celsius using liquid nitrogen. That's how we prevent the germination or wastage of the product or seeds or the genes. So it is useful for suitable of orthodox seeds or which will survive very long drying season. And so cryopreservation is method where ice forming ice, extracellular ice and intracellular ice and dehydration. And where this cryopreservation, one of the demerit is where the species may lack genetical adaptation and mutation, which would allow to thrive in its ever changing natural environment. But one of the another major demerit is these methods are ex situ conservation, gene bank and cryopreservation, everything are often involves money, which is often costly, and it is not suitable for all species. It can be done only for selected species. So this community seed bank, the community seed banks are seed bank which are available in the local community itself, where they can store the different types of products or crop products what they grow in their local storage facility with certain things which can be used as a sealed containers, and event of a disaster it can be used, resourced and reused. So genetical gene bank or conservation of genetical resources and store mainly reproductive samples. Also mainly they store variety of crops throughout the world, and these resources, whether they are from the farmers or breeding labs and research institutions. So the field of gene banking, these are the importance of field of gene bank. And if you go for the botanical garden or arboreta, so which are developed by the ancient British period, this botanical garden, so where we have important species available in this region, and where they maintain that species, where they can be propagated if needed, and they are mostly located in its in its natural environment, but in an ex situ conservation site in the form of botanical garden or arboreta. And Here we can see certain and important and botanical gardens or research centers for ex situ conservation. So, which are for Flora and Fauna International, and here are the national level institutions. So, Indian Botanical Garden, which is located at the Calcutta, and FRI, BS Botanical Survey of India, are few renowned organizations and which do conservation on the floral aspects. So, next one is the biotechnological ex situ conservation strategy. So, in vitro conservation and mode of propagation. So, clonal propagation, somatic and organogenesis and calculus differentiation. These are some of the methods or techniques of animals where we use the cryopreservation methods as where we go for interspecific pregnancy and aquaria. So, aquatic organisms we do aquaculture, and zoological gardens we call it as zoos, where we go and see lot of wild animals which are from our country as well as from other countries, where they are kept in an enclosed environment. So, like as I said, this cryopreservation also applies for animals, like storing of eggs, live sperm, embryos in the form of facility, and implanting embryos of an interspecific pregnancy is where implanting embryos of a species into the womb of a female of a sub-related species, not a different species, so related species, maybe a subspecies. So, aquaria research and public information which causes education, environmental awareness and education to all, and zoological gardens. So, zoos they also do certain activity like captive breeding programs. They are done in certain parts where they captively breed and release the animal into the wild. And what are the risks? So, mainly this ex situ conservation, the overall risk mainly depends on the transmission of diseases and resistance to the environment, because as they are grown in an artificial environment where protection is given by us. So, where it may lead to the genetical integrity among the population. So, the major drawback, it cannot recreate a total habitat ex situ conservation, where it can try to reintroduce a certain species into an environment, but natural evolution won't be happening. It is altered, so and it becomes one of the costly mechanism, because it needs lot of R&D, research and development, and continuous monitoring of these things, so that, and ineffective. So, sometimes it can be ineffective, even if you spend a huge amount of money, it can be ineffective, and their propagation in the wild becomes one of the major issues or question mark, and they are intolerant to diseases and pests, because as they have been in a lab condition or a controlled environmental condition, they have this lack. And other ex situ conservation strategies is one for the wild animals, translocation and release programs. It has been done for many animals like tigers or carnivore animals and certain herbivores. When the population is exceeded in the environment, it is captured and relocated to the different environment. So, with this I would like to come to an end of this ex situ conservation. Thank you for listening. Thank you.